Hello everyone, my name is Marlene Marquez. The objective of this video is to present my biochemical project for organic chemistry second semester. More than a year ago, the entire world was informed of the spread of a highly infectious virus, which now we know as COVID-19. When initially was believed to be a 2-week quarantine, turned into an entire year of social distancing, but also a race to contain the spread of the virus and create a vaccine. And how to forget, also a race to buy as much toilet paper as possible. This biochemical project is based on recent research related to COVID-19. I'll be analyzing the code N37BYJ from the protein database website. As you could see over here, the name of this file is crystal structure of SARS-CoV-2 antibody with RBD. This is classified as a viral protein on humans. The year is 2020. We're gonna be taking a closer look at the 3D view of the structure. This structure consists of 3 different chains. At the bottom, the purple and orange chain are the FAB fragments of the antibody. Over here, we have our heavy chain, and our light chain. And over here at the top, we have the spike protein or the antigen's epitope. As you could see, this molecule or this structure, should I say, looks pretty simple. We don't have a lot of ribbons going on, a lot of chains going on. If we look at it like this, we could see the interface over here. Here, we look from another angle. We see the space between the FAB fragments and the spike protein or the viral protein. And we're also going to be analyzing this structure using Chimera. As you can see over here, this is how the structure first looks when you open it up on Chimera. It's very simple. It kind of just looks like a breadstick. There's not much going on. Everything is just like this brownish color. We just have the ligand over here chilling. However, with Chimera, you could do different customizations or color codings. As you could see over here, I have color coded the structure by chains. And once again, you see the FAB fragment part of the antibody and you see the viral protein. If you move it around, you could see the interface or how the area in which they're interacting with each other. As of now, I have not done any simplifications or gotten rid or hidden any parts of the structure since it's still pretty simple. Now I'm gonna show you over here. I have color coded by secondary structure. The red areas are helix, the purple areas are strands, and the gray parts are coils. Now we're gonna be taking a look at this kind of interface. So over here, what you see is the area of interaction between the antibody part and the viral protein part. This area in red means that the residue in that area is interacting closer together than the residue that it's in the bluish area. Everything that is shown on balls and sticks are basically interacting. Now for the next part, I'm going to be showing you this same thing used without the interface barrier, so that you could see that everything that it's highlighted, the residue that is highlighted, it's what's playing part in the interaction. You could see some of it's right here, and this is a little bit farther. Over here, we can still see our interface area right here. Now I have gotten rid of a lot of the ribbons and the chains, or everything that was basically no playing part on the interaction. Over here, I'm going to show the interface color coded by residue type. So for the green part, it's all our hydrophobic area. The yellow will be our non-polar area. Blue areas are positive and red areas are negative. So we're gonna be discussing some of the interactions that are happening right here in this interface. First, I would like to mention vulnerable forces or dipole moments that are created between negative and positive areas. You can see we have a few of those over here in the middle, over here at the top, in this corner over here. Another type of interaction that is going on is, and that we see a lot in this specific structure, is hydrophobic interaction between amino acids. So if we place this structure like this, remember this is where we had our gap between our antibody part and our antigen part. And as you could see right here in the middle, all this is green, which means it's hydrophobic, and it's mostly surrounded by yellow, which is non-polar. So this is basically showing hydrophobic interaction among non-polar amino acids. And it makes a lot of sense that the green area, which is hydrophobic, will be surrounded by the yellow area that it's non-polar, because as we remember, hydrophobic doesn't like water, water is polar, so it makes a lot of sense that it's surrounded by non-polar stuff. Another kind of interactions that we have, if we get a little closer, are hydrogen bonds. I try to highlight the hydrogen bonds with these little blue lines that you can see over here. We have 3 of them just hanging around. We have another one right here, this over here. As you could see in this big gap, that was what was basically on dark blue on previous slide, so previous structure that I showed you was where it was further apart, and basically the only interaction that's mostly happening over here is a hydrophobic interaction. Here we see more hydrogen bonding. Now what I'm gonna do, I'm going to hide this interface so that we take a look at other kind of interactions. Other kind of interactions that I found on my structure are pi stacking. As you can see over here, this aromatic ring and the one below it are doing pi stacking. I think this one will be considered as D-shaped. Something that I do have to mention is I don't know if the distance between them it's enough to actually be causing that interaction. I don't know if they're close enough. here too, we could see by stacking interactions. Over here, you could see another T-shaped pie stacking. Now we're gonna go back to what I mentioned previously about FAB fragments. So what FAB fragment is, it stands for antigen binding fragment. This is a region on an antibody that binds to antigens. It's a molecule or molecular structure that can be bound by an antigen-specific antibody or B cell, which is an antigen receptor. It is composed of one constant and one variable domain of each the heavy and the light chain. And as if you remember, we have over here on orange our light chain, and here in purple our heavy chain. The variable domain contains the paratope, which is the antigen binding site, comprising a set of complementary determining regions at the amino acid terminal end of the monomer. Each arm of the Y-shaped antibody thus binds an epitope on the antigen. There are two ways to create FAB fragments. One of them is using the enzyme papain to cleave an immunoglobulin monomer into two FAB fragments and a FAC fragment, as you can see over here at the top. And the other way is to use the enzyme pepsin, which cleaves below the hinge region, resulting in a FAB prime 2 fragment and a PFC prime fragment. For the structure that I had decided that the FAB fragment shown that I'm analyzing appears to have originated from the use of papain, because if it had originated from pepsin, it would show an FAB prime 2 fragment composed of two disulfide connected FAB units. And if you could see over here at the bottom of my structure, we don't have any interactions between the end of the chains. Here you go. You can see that the purple chain and the orange one have no type of interactions. They don't have a bridge between them. Also, another thing that we'll be discussing is if someone was infected, in this case with COVID-19, their body will detect the foreign substance or antigen. To eliminate the antigen, the immune system starts producing antibodies. Antibodies are produced by B cells. Antigen binds to them and they create clones. Antibodies attack antigens by binding to them, and they either change the chemical composition of the virus, prevent the virus from attacking or penetrating body cells, or burst the invading virus, as you can see in this animation right here. Antibodies can fight viral invaders in two ways. Antibodies coat the virus, or macrophage, with which they destroy it. We're gonna also be talking about ELISA testing. ELISA stands for Enzyme Link Immunoabsorbent Assay, and these tests are used for diagnostic purposes. There are four different types of ELISAs: direct, indirect, sandwich, and competition slash inhibition. A little backstory. Back in the summer, my mom contracted COVID-19, and she did test positive. She did got tested. However, nobody else in my house got tested since we assumed that we're in close contact, direct contact with her. We were most likely to also have the virus. So we just decided to quarantine with her. Therefore, if I wanted to know if I also had the virus or no back in the summer, I would do an ELISA test to check for antibodies. The ELISA test that I will choose to use is indirect. The indirect ELISA is suitable for determining a total antibody concentration in samples or if there's antibodies. To do an indirect ELISA, first a microturtle well is coated with an antigen. A coating buffer is added to help bind the antigens to the well, and they cannot be removed. Then the sample, which in this case will be my blood, is added to the well. If the sample contains antibodies, it will bind to the antigen. Then any unbound antibodies are removed using a washing buffer at least 3 times. Then a chromogenic substrate is added, which gives color to the test if the test is positive. Therefore, if there's color in the test that I will be doing, that means I did have the virus because there are some antibodies right there. And the more intense the color, the higher the concentration of antibodies. And this is it for my project, for my presentation. Thank you so much for watching.